and then we will go directly into the first lecture, which will be myself. It was planned to be Professor Manuel Carondo from Portugal, who usually gives the first talk, but this year he could not make it to come to Rio, so it was the nightmare for me, because for an organizer to give the first lecture, the organizer sleeps 4 hours just in the night to finish the slides. So just to remember that Rio, because we were a capital city in Brazil, we have lots of scientific institutions, so we are a science hub in Brazil. All red bullets are scientific institutions, and in the university island here, the UFRJ main campus, there are many other scientific institutions other than UFRJ in the Technological Park, the Research Center of Petrobras, the Research Center of Eletrobras, CETEM, and many others. UFRJ is a huge university. It rotates among the top 3 universities in Brazil, together with Unicamp and University of São Paulo. We have many graduate and undergraduate students, more than 60,000. And COPPE is a coordination of 13 engineering graduate and research programs. It's 62 years old now, and it holds a top position in research in engineering sciences in Brazil and Latin America. We are in COPPE, we have decades of experience of interacting with industry through COPPETEC Foundation. So our lab is the Cell Culture Engineering Lab of COPPE, and we are, for over 20 years now, dedicated to the development of technologies for the production of biologics, focusing on those that are derived from animal cell culture. And the current projects, they focus on recombinant vaccines and monoclonal antibodies, as you see in the transmission electronic microscopy pictures that we put here. In the past, we have had collaborations with Professor Stephen Hayen from UFRJ, with Professor Amin Kamen from Canada, on other areas, for example, stem cell scale-up, and also production of viruses with biomanguinhos and viral vectors with Canada. So all those areas rely a lot on animal cell culture, so this is the focus of what we do at LEC. You see here some pictures from our lab, from our culture rooms, and also from the purification sector. And here the clean rooms where we did scale up. For example, during COVID pandemics, in late 2020, we were doing cultivations at 35-liter scale and purifications at 25-liter scale using the ActaPilot system. We started the lab doing mostly cell culture and purification. Before the lab was created, the team had already worked with perfusion cultivation, so continuous biomanufacturing, which is a trend in the last 10 years, was already what we focused on 25 years ago, using different cell retention devices like the ATF, hydrocyclone, spin filters, rotofilters, and trying to develop perfusion cultures that use the minimum amount possible of culture media. But we had, right at the beginning, to start doing cell line development, to have interesting cell lines to work with. And we learned in the last years to do, in a more regulatorily friendly sense, cell bank construction and also the certification of cell banks together with partners that we selected. And it was also in the last years that we did also the scale-up of the purification processes, as you saw in the last slide. So this is the experience we gained during COVID pandemics. So we had to scale up 33 times, 40 times upstream and downstream processes. We learned how to do animal studies for safety evaluation. outsourcing GLP toxicology studies, designing those studies, and also doing efficacy studies at the biosafety level 3 lab or animal facility of UFRJ and INCA. Also, we learned a lot how to do development and validation of analytical techniques for quality control and stability studies. And because at that time our clean rooms were not operational, we partnered with Bionovis. They allowed us to say for the production of the API. So we took the bioreactors from LAC and the ACTA pilot from LAC and the team from LAC and the raw materials from LAC, and we used the pilot plant of Bionovis to produce the API, and had a full support of Juliana, Wanda, and the QA, the quality assurance team of Bionovis, to produce our first clinical lot. And for this vaccine, we also learned to prepare dossiers and present them to Anvisa, and to present a pre-IND to FDA this year. And based on that experience, we are implementing now the biologics pilot plant of COPPE, UFRJ. Sorry, this is old. This year we had like renovations. Sorry, it was like during the night that I prepared those slides. So since June this year, our clean rooms have been certified as Class B and Class C. We had changes in the HVAC system because we wanted to have Class B rooms in order to be able to do the filling. So with laminar flow hood Class A inside a Class B. So after this kind of renovation, this revamp that we did this year, were able in June this year to certify our clean rooms as Class C and Class B. So our pilot plant, we are planning to do to provide as a service or as a partnership through COPPE Foundation, scientific advice and support, process development for cell line development, upstream processes, downstream processes, scale-up, analytical development and validation, stability studies, both long-term and accelerated, cell bank production, and designing the strategy for certification, and also producing clinical phase 1 drug substance and drug product lots, as we did for our vaccine. And we are just preparing, because of the course, we didn't do it in the second semester. We had to postpone for Q1, Q2, 2026. We will produce already inside LAC this time, not using the clean rooms of others. Now we have our own clean rooms operational. So we would produce our second clinical lot of UFRJ VAC, our vaccine candidate, which is both intramuscular, is being developed to be intramuscular, and in another development to be intranasal. So we will be producing inside those clean rooms by the beginning of next year. And also, we can help also with documentation, preparing analysis forms, preparing batch records, preparing for Anvisa the DDCM, and preparing for FDA the pre-IND. It's everything that we learned in the last years. And now regarding this course, it's the 9th edition of the International School on Production of Biologics. Everything started in 2004. Some of our speakers are here. Some of our students of 2004 are nowadays speakers. Some of them are laughing. So you will see some of the people here in this old picture. So 2006, the second edition, 2008, 2011, 2013, 2015. So every 2 years, at some moment we switch it to the uneven years. Because in the even years there is the Latin American Conference for Animal Cell Technology, so we switch it to the uneven years so that the Latin American community can meet every year. This is 2017, 2019, but then pandemics came and we got pretty busy with vaccine development, regulatory issues, renovation of clean rooms and everything. So It was a 6 years of interruption, and now we are here again for the 9th edition. So last edition was in 2019, and now we have the 9th edition. Including this edition, it's about 1,200 people that have been trained in this course. So it's quite a lot, and it's very interesting that every time I meet people, people say, oh, I was in one of your international schools. Obviously, I don't remember all the 1,200 people that have been here, but we have had people from all continents here, both as speakers or also as participants. We had people from Australia, from Turkey, from Tunisia, from Germany, from Sudan, from Nigeria. All those people have already been in the different editions here. And our program this year is very exciting with great speakers. So I wanted to thank all of our speakers to be here, to travel, to here, to be with us this week. We'll be starting in 5 minutes with the first lecture, which will be by myself. So I forgot to say my name. I'm Leda Castillo, professor of UFRJ. At LEC, we have also Professor Renata Alvim and Professor Rui. You will have also the opportunity of having lectures and workshops with them. And we'll have every day from 8:30 to 3:15 p.m. We will have lectures in this room. And every day from Monday to Thursday, we will have workshops. So the workshops are intended to be in a smaller group and more interactive. And those workshops will take place in different rooms. So one here, one on the other side, another a bit far away on the other side of the corridor, and the last in the end of the corridor. But the team, our organization team and the speakers have this different badge. So everybody that is noted organization here, you can ask them for help. And on Friday, instead of workshops, we put together a roundtable with that will be very, very interesting about the biopharmaceutical industry in Brazil. It's an interesting story that started around 2006, 2007 with people from the Brazilian Development Bank noting that there was a big deficit in the Brazilian trade, and it was mostly in the health area, and within the health area, the biologics were the guilty for that. So the federal government at that moment initiated studies and measures and laws to try to develop the biopharmaceutical sector in Brazil. And nowadays, we have several companies, both public and private, producing biologic APIs in Brazil. And biologic APIs are much more complex than synthetic APIs. And if we look at the synthetic drugs in Brazil, they are mostly imported, or the API is imported from China, from India, or the whole drug, the finished product, is imported. So it's very, very good that we can say nowadays that we have biological APIs being produced in Brazil. They are much more complex than synthetic drugs, and we are in a better position than maybe we are in the synthetic drugs. In terms of health sovereignty, what we saw during pandemics, that's very important. If we don't produce APIs, maybe the planes will be stopped in the connecting airport as the API vaccine. So there were stories of API not leaving India to come to Butantan to produce Coronavac. There was story of a plane with API stopping for refueling in the US and not being allowed to depart, and our people without vaccines, and 400,000 dying in the first semester of 2021, when in other countries people were already being vaccinated and were protected. So health sovereignty is something that we learned from COVID pandemics that is very important. So it's very important to be able to develop new products and new processes in our countries and to be able to react to the future pandemics in a more autonomous way. So, housekeeping notes to finish with this part. For the workshops, we have rooms of different sizes. So we need to know in advance how many people are going to each workshop. We will have 3 workshops in parallel. Today, tomorrow, and on Thursday. And on Wednesday, we will have 4 in parallel. So please, until the end of lunchtime, which is 2 p.m., go to the registration desk and sign up in a paper for which workshop you want to go. And then around 4 p.m., we will have signs around the registration desk saying in which room, which workshop will take part. Certificates, we are an university, so we ask you to sign for attendance in each lecture, and we will only give certificates to those who have really been here. Otherwise, maybe you attend one lecture and then you say, I have been in the International School of UFRJ, and you don't know how to answer any question that people pose to you in an interview, in a job interview, and then it's our name that is bad. So, as in all classes at universities, we will ask for attendance. So please sign the lists that will go through the seats during each lecture. For participants, today and Thursday and Friday, please go on an individual basis to have lunch. There are restaurants in the back, trailers in the front. But Tuesday and Wednesday, we have a very interesting, very nice thing to offer to you. We will have, in the first half of lunchtime, we will have on Tuesday a presentation by Merck, a very interesting presentation on cell line development, I think. And on Wednesday, we'll have in the first half of lunchtime, we'll have Kuhner presentation about shakers. And because we will have just 45 minutes to have lunch, those people who are attending to the Merck and to Kuhner presentation will be invited to go out here, to go to our tenda, and we will offer you a lunch. So Tuesday and Wednesday, please stay here for the nice presentations and then go to have lunch with us here. And for the speakers, I ask all the speakers to come here to the front when lunchtime started, so that we go together to a restaurant here in the campus. Okay? So this is what we had as an outline of the course. If somebody has some question about the course, yes? No, it's one per day. So there are 3. You sign up in one of the 3 lists. It's the first come, first served basis. So go during coffee break at 11 p.m., go there and already sign your name, because if you go there and there are already 50 people in one workshop, it will be closed, because it will be for the other 2 workshops or other 3 workshops on Wednesday, okay? Just one per day. Good question. So he asked if it's just one workshop to choose from every day, so it's just one. So please go to the registration desk during the coffee break or during lunchtime, not later than 2 p.m., which is the end of lunchtime, to sign up for the one workshop you prefer for that day. Okay, so let's go now to start the scientific program. I have now 45 minutes, right? No, okay, thank you. So this lecture is intended to be an introduction on biologics and animal cell culture. And when we think of biologics, we know that they can be used for therapy, for prevention, or for diagnostics. So biopharmaceuticals, biotherapeutics, like recombinant proteins, including monoclonal antibodies, or also recombinant hormones, recombinant coagulation factors, recombinant enzymes, and many others. Also, advanced therapies like cell therapies, gene therapies, or combinations of cell and gene therapies. All of them belong to this class of products for therapy. There are also the vaccines, which are the oldest class of biologics. Vaccines are intended for prevention. We have seen a lot of development in the vaccine field over the last 2 decades, and this is very good, because otherwise, without the huge development in vaccinology over the last 2 decades, we would not have had a vaccine in less than 1 year to fight the COVID pandemics. And diagnostics, we can have in vivo or in vitro diagnostic tools, and most of them rely also on biologic consumables or raw materials. One thing that I forgot in the housekeeping notes, we had even planned to do signs for the walls, but we didn't do. We ask, as in all international conferences, we ask you not to take pictures of all presentations. It's not myself, but usually in all international conferences, we ask people not to take pictures of posters, not to take pictures of presentations. So please, if you want any information from any speaker, go after the presentation in the coffee break and ask the person for that. So if we look in those classes, we can have vaccines of different types, vaccines based on recombinant proteins, recombinant virus-like particles, on the viruses themselves, or virus vectors carrying the gene of some other antigen, vaccines based on nucleic acids, here a DNA molecule, but also, as we saw, a huge development in mRNA vaccines that we will discuss today in the afternoon. Therapeutic proteins, many people put MABs, monoclonal antibodies, separately because they are so important in terms of market, and we will see that. But we must remember that antibodies are proteins, and if those monoclonal antibodies are used for therapy, they are biotherapeutics. They are therapeutic proteins, so they should be together with also other therapeutic proteins. Many people call them biopharmaceuticals. We have therapies based on nucleic acids, DNA, gene therapies based on DNA, or therapies based on RNA, cell therapies, many of them based on stem cells or cells derived from stem cells. And also there is a big mix or a big combination between gene and cell therapies. And that's why they already maybe 10 years ago or even a bit more, the European Medicines Agency has created a term ATMP, Advanced Therapeutic Medical or medicinal products, not medical products, medicinal products, ATMPs. And about 5 years ago, for example, in Brazil, 3 to 5 years ago, Anvisa in Brazil has made new regulations specific for advanced therapies. For example, CAR T cell therapies are included in advanced therapies. Diagnostic products, if they are for use in vitro, for example, producing a monoclonal antibody to be the main reagent of a diagnostic kit, of a ELISA kit, or a rapid test kit, or maybe an antigen, recombinant protein to be used as an antigen in an antigen test to detect infection, for example. This is an in vitro test, and many of the main reagents of that kit, of those kits, are biological products. But there are also biological products that are used for in vivo diagnostics. So the requirements for quality of these products are very similar to those of biotherapeutics or vaccines, because they are injectable. And if you are injecting in a human person, purity requirements tend to be very, very high. And there is a class of products that is in development. Many people developing some products already in clinical trials, but I think no approved product so far, which are EVs, extracellular vesicles, including exosomes, which is one type of EVs. So there are several clinical trials, and so let's keep an eye on them. Maybe they will be next to be the first product of the class to be approved. Regarding market aspects, the biopharmaceutical industry has been increasing in market size over the years. So here we see from 2019 up to the predictions for 2027, but we see a steady increase in, if we sum up all four, so here monoclonal antibodies in yellow, cell therapy in orange, recombinant hormone and other recombinant proteins other than monoclonal antibodies in gray, and other types of biologics in blue. If we add all those, we see a very consistent growth in all classes, and monoclonal antibodies dominating the market. One single class of recombinant proteins being responsible for a very large part of the market. We see here also in blue that monoclonal antibodies are individually the main class of product. This is interesting. This is regarding monoclonal antibodies only. So this is mAb approvals as a percentage of total approvals. So we see in the last 10 years, since 2015, we see that more than half of the new biologics being approved were mAbs. So this is very impressive. And we see mAb sales as a percentage of total sales. So new products over 50%, but 80% of sales, of total sales of biologics. So 80% of total sales of biologics are from monoclonal antibodies. So that's why everybody, every company wants to develop monoclonal antibodies. Even when just 10 to 20% of the new approvals were monoclonal antibodies, they represented already around 50% of the sales. And this is the global biologics market by application in 2020. Immunology, oncology, autoimmune diseases, and infectious diseases. And here, this is also from Walsh's review. So Gary Walsh from Ireland publishes every 5 years. So next year we can anticipate that he will be publishing his new version of the review, always in Nature Biotechnology. So this is his latest review. Those reviews are always very interesting. So he, in those plots, he looked at the European and US approvals in the indicated periods here per year in the 4 years before his review, so 2018 to 2021. And here he takes since the 80s, the 1980s. So we see here that in those 5 years, in those 4 years, we have continuously large numbers of biologics being approved, around 30, let's say, in average per year in the EU and US. Regarding new approvals, he included genuinely new biopharmaceuticals. So from adding up those 4 years, from 2018 to June 2020, when he published, there were 85 genuinely new biopharmaceuticals being approved in the US and in Europe. 58 biosimilars, which are biologics with expired patents. Reformulated Me2 different indications and related products, so not a genuinely new. 31 new products. And previously approved in another region of the world that is not US and Europe, 15. And interesting, if we look in the different 4-year periods that he shows here, we see that from 2015 to 2019, we see a huge increase in new approvals. And I guess he didn't include here 2018 to 2021 or 2022 in this plot because pandemics messed a little bit everything with many COVID vaccines being approved at once. So that's probably because he didn't, the reason why he didn't include here 2018 to, or 2020 to 2022. This is also from his review. During pandemics, this refers to 2021, the 20 top selling biopharmaceutical products. We see, obviously, in 2021, the Pfizer-BioNTech vaccine as the top sold product, $37 billion in just 1 year of sales. But the second top product is adalimumab, Humira, $21 billion. The third is also an mRNA vaccine. So mRNA vaccine from Pfizer-BioNTech, mRNA vaccine from Moderna, almost $18 billion. But then you see many mAbs, pembrolizumab, ustekinumab, nivolumab, another mAb, 2 combined mAbs, mAb-derived, mAb. So we see in the top 20 products how important the monoclonal antibodies are. And if we take out the COVID vaccines, the top ones would be all mAbs. If we look at the data before pandemics, how was it? The 10 top sold biologic products before pandemics, this is data from 2019, we see that 7 out of 10 were biologics. And from those 7, we see mAb, and this is mAb-derived because it's a fusion protein. So you see how important the monoclonal antibodies are also in this way. And we see here about $20 billion for Humira in 2019, and 2021 it was 21 billion US dollars. So still increasing, although Humira is already for many years as the top biological drug in market size. So it's still increasing. But many biological products have lost already patents. So the first patent of a biologic drug to expire was erythropoietin in 2004. So the first biosimilar epoetin-based products were approved around 2007. First approvals were in Europe because Europe was the first agency to prepare at that time new regulations for products with expired patents. And it was also Europe that created this term biosimilars, not biogenerics, because the products are so complex that you cannot do just a comparability study or bioequivalent study as you do for synthetic generics. So Europe created in their regulations from 2004, they created this term biosimilars that is nowadays used everywhere in the world. So the first epoetin-based biosimilars were approved in 2007. Human growth hormone is a very simple protein, so it's kind of an exception of the minor amount of products that can be produced by bacteria. So it was biosimilar human growth hormone-based products were approved first. But if we look, it was here map-based and related. It was starting 2013 that we had approvals of biosimilar mAbs, and nowadays we have lots of them. So, for example, adalimumab-based, the competitors to Humira, we have a lot nowadays. You see here the reference product here is Humira. Here reference product is Remicade for infliximab, rituximab, trastuzumab, bevacizumab, and others, also fusion proteins like etanercept. So we have nowadays many different biosimilars approved around the world and also in Brazil, also in Latin America. And we must say that before the regulations came, some countries in Latin America had already developed biologics, and our Argentinian colleagues from Santa Fe, they were already producing recombinant erythropoietin in the 90s. And they still export, I think, to Brazil the API for Brazilian products to be produced with their API. Cuba had already developed also erythropoietin and monoclonal antibodies around the 2000s. And also Mexico. So we must say that there are three countries in Latin America that were pioneering in that area when we in Brazil were not. So regarding the development of a biopharmaceutical product, let me just check the time. Regarding the development of a biopharmaceutical product, it's usually a very long way and a very tough way to go. And after discovery of a molecule, the people in the biomedical sciences area, they start doing animal assays to check if this is promising or not. The biochemical engineers and chemical engineers like us start developing processes, developing recombinant cell lines and cell cultivation processes, purification processes, process development until we have a very productive process that that is economically feasible, because otherwise you will not go to market if it's not economically feasible. So a very good process development, which must start from a very good cell line. So this will be the two talks on Wednesday morning by Professor Marcelo Brígido and Professor Alan Dixon. A very good process starts with a very good cell line with a high specific production rate. So process development is occurring while preclinical studies are being done, and maybe you can further improve your process, your bioprocess, while phase 1 is occurring, and even maybe start of phase 2. But when phase 3 starts, you must already have your process very well defined and validated and characterized. Just after phase 3 clinical trials with a very large number of patients is concluded, that the dossiers with data for pharmaceutical quality, safety and efficacy is then submitted to the to the regulatory agencies like Anvisa or FDA or EMA, and if the regulatory agency grants approval, then the product can be commercialized. So it's a highly regulated industry. All must follow good practices, and quality assurance is very, very important, because the products are so complex that their quality is highly dictated by the quality of our process, by the reproducibility of our process, which in turn has as a consequence the reproducibility of our lots, of our batches. So this is a long way to go. In normal times, this takes maybe 10 years. For vaccines, it can take 20 years, because for a vaccine, many vaccines are developed, but then outbreak is gone, and you don't have patients to do phase 2 and mainly phase 3. And for a vaccine which is to be used in the whole population, sometimes the clinical trials must have 40,000, 50,000, up to 60,000 people have already been enrolled in clinical trials of vaccines before the pandemics. And in COVID pandemics, we had 3 things, in my opinion, that were able to accelerate the development of vaccines without compromising quality and everything. So nothing was compromised. All the tests were done. So we have a huge development of vaccinology in the 2 decades before COVID pandemics, for good luck for everybody of us. So there were already mRNA vaccines in clinical phases, in clinical studies, starting just a few years before pandemics. So the viral vectored vaccines, for example, based on adenoviruses, like the the Oxford vaccine, there were already vaccines for other applications, including other coronaviruses, in clinical tests. Many mRNA products were being tested mainly for cancer therapy. So BioNTech in Germany, which is the source of Pfizer, BioNTech vaccine was mainly focused on cancer therapies, mRNA therapies. So this is one point. The other point is that there was investment. Which investment? So it didn't depend on the administration boards of the companies to decide will we do the next investment? No, the countries were buying in advance hundreds of thousands of doses. So with that money, the companies were doing everything in parallel. Everything that had to be done was done in parallel and not sequentially, because there was money for that. Usually there is not so much money, so everything is done in steps, and one step is just approved by the board of the company if the last studies, phase 1 study is very promising, so let's do the investment for a phase 2 and so on. So the availability of money, of investment, of some countries buying in advance the vaccine doses, this helped a lot, developing more quickly the vaccines. And the third thing is that because the development was so quick, it was possible to do the clinical trials, including the phase 3, while the virus was still circulating and circulating a lot. So you could enroll 40,000 people for Oxford AstraZeneca, 40,000 people for CoronaVac, 40,000 people for for Pfizer, and for all vaccines you were able. So I was a volunteer in a Chinese vaccine that is approved in China nowadays, but was not approved, was not submitted here in Brazil, but the clinical trial was done in Brazil. So many people, and I heard in many vaccine conferences that people are grateful to Brazil because we had so many people volunteering here for the clinical trials. And maybe here the virus was circulating so much because of other reasons that we had so many people wanting to be volunteers as well. So this enrolling so many people in such a short time allowed to conclude each clinical trial much more quickly and gathering the data to present a BLA. BLA is a biologics license application, and the regulatory agencies were also doing all the evaluation in real time in order to have no delays. So the development of a biological product or of new medicines in general, this is also for synthetics the case, is not only long, but it's also like a small probability that what is starting to be developed there with basic research, drug discovery, that it happens to be an approved medicine several years later. Because there are many failures here. So you can already, in the preclinical studies in animals, see that it's not safe or it's not efficacious, and you stop development. When maybe, let's estimate here, 2/3 of the products go to phase 1, but in phase 1, which is evaluating safety in human volunteers, you have this big funnel here. So maybe just 1/3 or 1/2, maybe 40% of those that enter, of those products that enter phase 1 have safety profiles that allow them to continue to phase 2. Phase 2 is the first time that therapeutic agents are evaluated in patients who have the condition. So we have a big funnel here as well, and then in phase 3 you test patients in a large scale to have a statistical confidence. And that's why we see that big funnel. So usually pharmaceutical companies, they put on this one product that is approved, they put all the costs of all the other molecules that started development and did not make it to commercialization. So that's why we see, for example, in associations, industry associations like this one in the US, they put here the average R&D cost required to bring a new FDA-approved medicine to patients is estimated to be $2.6 billion US dollars over the past decade. But we are speaking about biologics, and we have seen already earlier in this lecture that those products are large and complex products: proteins, nucleic acids, viruses, virus-like particles, extracellular vesicles, or the cells themselves. So they are difficult to produce. And if we compare, for example, a monoclonal antibody to aspirin pharmaceutical, active pharmaceutical ingredient, we see when we put in the same scale how much more complex and larger the monoclonal antibody is. If we look at the molecular mass, 180 Dalton, and here molecular mass 146,000 Dalton. And here you have also post-translation modifications and many other things that make this product much more complex. And that's why those complex products require complex production processes based on complex expression systems. And that's why higher eukaryotic cells are the main expression systems to produce recombinant biologics. And one of the main reasons for that, and this will be the talk of Professor Laura Palomares from Mexico on Thursday morning, are the post-translational modifications, or PTMs. So I give as an example, just very shortly, human coagulation factor 9, because this protein suffers almost all possible post-translation modifications, so carboxylation, hydroxylation, glycosylation, sulfation, phosphorylation, activation peptide. So many different post-translational modifications. So probably only a mammalian cell will be able to do that, to do those post-translation modifications in the correct way so that the recombinant protein is biologically active. And that's why we see here in blue the percent of the products that are produced by mammalian cells and the percent that is produced by non-mammalian cells. So in the 80s, the very few first biologic products that were approved in the 80s, 2/3 were produced in bacteria. So for example, human growth hormone and GCSF, for example. And just 1/3, this was TPA, produced in CHO cells. But with time, we have seen that more and more mammalian cells have started to dominate. And in the last years, we see about 70% to 30%. So around 70% of the products are produced in mammalian cells. So we saw that those complex products require complex production technologies because most of the biologics are either recombinant proteins or genetically modified cells or nucleic acids or viral vectors. Mostly we use recombinant DNA technology and hybridoma technology, technologies that were developed in the 70s. And they enabled the development in the 1980s of recombinant biologics. Also, for recombinant monoclonal antibodies, it's common to have a combination to use hybridoma technology and then recombinant DNA technologies. We want to produce molecules that are complex, they are large, they suffer several post-translation modifications that are important for biological activity, for stability, and to avoid, in the case of therapeutics, to avoid immunogenicity. And that's why we saw in the last slide around 72% of the biologics are produced in mammalian cells. If we take animal cells, then this number rises to about 80%. And so that's why the focus of this course is on biologics produced by animal cells. I'll very shortly speak about some historical milestones that allowed the in vitro culture of animal cells. It was by the end of the 19th century and the start of the 20th century that some developments were done that gave the basis for us to be able to cultivate cells from an organism in vitro. So Roux was able to maintain embryonic chick cells in saline solution. Harrison grew frogs' nerve cells in vitro using clotted lymph fluid as source of nutrients. It was Harrison that realized that the cells require a solid support because most of the cells are in solid organs. And something that is so obvious for us nowadays, but it was he that noted that, cells require nutrients to proliferate. Roux and Jones used trypsin for the subculture of those cells that adhere to a solid support. And Carrel developed aseptic techniques, including the Carrel flask that we see here. That was the origin of the tea flasks, of the cell culture flasks that we use today, made of plastics. Between the 40s and the 60s, Enders grew poliovirus on cultured human cells, and this was the origin of developing viral vaccines. It was at that moment, around those days, that important cell lines were established, like the Chinese hamster ovary cells, CHO, baby hamster kidney cells, BHK, and Vero cells, and HeLa. And Ego published a key study on amino acids metabolism, trying to propose defined culture media. And Hayflick and Moorhead showed that human cells have a finite lifespan. In the 70s, we had the start of modern biotechnology because of development by Köhler and Milstein of hybridoma technology, and also the development of recombinant DNA technology was established. And this happened in the 70s and allowed then that in the following decade, recombinant insulin was approved for human use. So the first recombinant protein to be approved for human use was in 1982, but insulin is a very simple protein, so it can be produced in E. coli. A monoclonal antibody was licensed for therapeutic use, OKT3, and recombinant TPA, produced in Chinese hamster ovary cells, was approved in 1986. So this was the first cell culture-derived biotherapeutics to be approved. In the 90s and in the first decade of the century, humanized antibodies were licensed for human use, and this allowed many more monoclonal antibodies to be approved. Human embryonic stem cells were isolated, and induced pluripotent stem cells, IPS, were developed. The first gene therapy was approved for human use in China in 2003. First biotherapeutics produced in transgenic animals were approved in the US in 2009. In 1998, the first antisense therapy blocking specific mRNA was approved. And the first off-patent biosimilars were approved after the regulatory pathway definition. In the last 2 decades, the first biotherapeutics produced in plant cells were approved in 2012. Gene therapy was approved in 2012, many years after Gendicine. Other gene therapies have so far been approved as well. Several antisense therapies blocking specific mRNAs were approved. The first biosimilar mAbs were approved. In 2014, the first viral vectored vaccine was approved for Ebola, and this was the basis for the development of viral vectored vaccines that helped us so much as well in COVID, like the mRNA vaccines. So the Oxford AstraZeneca, the Gamaleya, the Janssen vaccines were based on viral vectors. So the approval of the Ebola viral vectored vaccine in 2014 was an important step. The first bispecific antibody was approved in 2014, and this is a great trend nowadays that Andrea will talk to us today in the afternoon. First, mRNA vaccines approved during pandemics, and CAR T cell therapies and several other types of advanced therapies were approved. And with that, I end my presentation. I think we have just a couple of minutes for questions. And I wanted to thank you for your attention and thank the people both the present people that are on the picture and that you see here around the conference room, but also our past students and collaborators. And also we so far have been just surviving with money from Brazilian funding agencies, so it's a tough way, but it has worked. So we must say CNPq, FAPURJ, and COPPE, without these agencies, our lab would not exist. So thank you so much.
So do we have any questions?
Okay, so we can move forward.
Yeah, yeah.
So, Leda,
A question?
I have a question. Just a provocation. Don't you think that after the pandemics with the mRNA-based vaccines, we have such a free pathway for doing this therapy based on mRNA? Don't you think it provokes the regulatory agencies and so on to be more, acceptable for those therapies?
Yeah, I agree. So safety for mRNA injection is already proved in millions of people. So I think this will accelerate that mRNA-based therapeutics will be approved more and more in the next years. And probably Patricia today in the afternoon will, before the lunch. Yeah, just before lunch we'll have the lecture by Patricia Neves from Fiocruz, and she will probably give some insights about that as well. But I do agree with you.
So thank you, Professor Leda, for your presentation. Here's a small gift from us. Thank you. So moving forward with our schedule, next presenter will be Guillermina Forno from Argentina. Guillermina has more than 20 years of experience in the biotech industry. She is R&D manager at Amega Biotech with experience in the development of both microbial and mammalian cell processes. She is also professor at Universidade Nacional del Litoral and coordinator of the bachelor degree in biotech in that university. So welcome, Guillermina.
Well, thank you very much. I will need something to... Perfect. Well, hello, thank you very much for the invitation, Leda. For me, it's a pleasure to be here with you. And well, I think that if you have any comment during the presentation, don't hesitate to do it, and we will talk about that. Well, why to talk about from nature to newborn products? Well, we are in a biopharmaceuticals evolution, but nowadays some paradigm shifts, as it was presented previously by Leda. I think it's very exciting for us to be here and to adapt ourselves to these shifts. Well, as Leda was presenting, we can say that the recombinant DNA production, the production of biopharmaceutical base in recombinant DNA started in the 70s with the production of insulin in bacterial hosts. Then we had our first biological that was approved in the 80s that demonstrated that it was clinically viable. Then after that, we had a tremendous growth of biotech industry with monoclonal antibodies and other complex biologics that transform the treatment of many diseases across different indications. And well, of course, it changed everything for medicine and for the treatment of many diseases. Nowadays, we are in a new era with the validation of the mRNA technology. After the pandemic, as it was presented, we know that this technology is capable to a rapid response. And nowadays, we can add the emergence of large sets of data that can be processed. We can take more advance of that data, and we can add artificial intelligence. We will have a presentation in this course about that. And well, who knows? Who knows in the next course, because everything is accelerated, and probably we will talk about more and more products being approved in a faster way in the near future. If we talk about paradigm shift, we can set the first shift when we passed from small molecules to complex biologics. Well, it was a big revolution, many doubts at the beginning, as used to be. Now we are talking about faster R&D and regulatory pathways that are changing, as you mentioned before, Andrea and Leda. I mean that regulatory agencies can be confident that with the data that we have today, we can emphasize safety and evidence generation, but not everything must to do in clinical trials. We have some other tools nowadays to be confident about the success of a new product. And an additional shift is the integration of advanced platforms that can contribute to create new therapeutics, more efficient, more flexible to treat some disease and to contribute for the treatment of some particular diseases that are unattended today. Well, as it was mentioned before, we have a big market for biopharmaceuticals. That growth, it is rely in the growth of population, but not only that, it's only the growth of diseases that are related with age. And of course, we have an aging population, and we have new diseases to be attended. And also the, the focus in diseases that are, maybe orphan diseases that must be attended to. Well, the market is nowadays dominated by monoclonal antibodies, and we will discuss why they are so successful. We have several factors, but in few words, we can say that they are highly efficient. They can treat several diseases, and we have nowadays a platform for producing it. It's quite easy to scale up a process. And for vaccines, that is a most opportunistic segment. Well, we have also a big opportunity with new modalities where we can have a new opportunity, taking into account that the emergence of new diseases requires very often a rapid response. We can see growing oncology pipelines that are the more interesting because of the revenue, but also cardiovascular diseases that are growing a lot and related probably with age, as it was mentioned before. With trying to find new drugs for treating those cardiovascular diseases, and the treatment or the opportunity for treatment of rare diseases too, across the world for different applications. We can add to that the need or the worry or the care of the states or the healthy payers of obtaining more affordable drugs. States are working in different points. One of these is to accelerate the biosimilar development. We have a quite new guidance from the FDA where it is very clear that probably clinical trials are not needed. I mean, it is written now. The FDA is the first time that put that in an official guidance. And also, well, the states are trying to put more effort in the process of acquiring drugs, taking into account the price, because it's very difficult, and I can see that in Argentina and probably in Brazil too, how difficult it is for payers to put all the money that they need for paying very expensive drugs, and well, at the end it's not affordable for many states, even for the bigger ones. Well, regarding monoclonal antibodies, of course, we have the FC regions and the FAB regions, where it's specific and it's the antigen binding site. Andrea will talk a little bit or better with in the next talk. And we will analyze now why have monoclonal antibodies been so successful. Well, the first point is the high clinical utility. They have an exceptional specificity in the FAB region. We can explain that in the structure of the molecule. They are used in a wide range of therapeutic areas. We can see it in every therapeutic area. And the pharmacokinetics is very, very predictable because of the FC region that gives to the antibody a very predictable pharmacokinetics. And here we have the antibodies. The first one started in the 80s, the first approved one, as Leda was presented. And here we have in red several antibodies that were the best-selling drugs in 2024. I mean, we have, in 2021, we had the monoclonal antibody number 100 to be approved in the FDI. I don't know exactly right now how many we have, but in that time we already had the 100 map approved at the United States. Well, you can see the market is really huge, $300 billion, and well, it's growing. And if we can see the indications, we will have, several kind of cancers, arthritis, Crohn's disease. Some of them has proved to be very successful because they can be used in several indications like pembrolizumab or nivolumab, for example. The other important factor that contributes for the success of the MAPs is the scalability and the robust manufacturing. We have nowadays a well-established production platform and standardized purification strategies from the use of the protein A that binds the antibody by the Fc region. We have a very predictable result in downstream processing. We have also clear regulatory pathways and supply chains that are very robust. We have several providers that give us a full set of solution for establishing a new manufacturing facility for monoclonal antibody. And here I am quite impressed to know that some companies, for example, Samsung Biologics, have the capacity to run fermentations in 600,000 liters. They are building nowadays the fifth plant. They will reach more than 700,000 liters. And here we can see the scales, the current scales. We have 15 liters bioreactors, several units. Apparently for them, this is the limit because even the plant 4 contains 15 liters bioreactor. However, some other companies say that they have 20 K liters bioreactor. We have also a shift, from the stainless steel technology to the single-use technology that provides flexibility because you don't have to do all the work that means cleaning validation from changing from one product to the other product. But at the end, you have that in the world we have a huge capacity for producing monoclonal antibodies. The other factor for the successful of these products is the advances in engineering and design, and Andrea will talk about that today. We have different possibilities of engineering the antigen binding sites or the Fc in order to obtain or to modify the properties of the antibody. And nowadays we know a lot about that, and therefore we can design a product that obtain the desired effect, the desired clinical effect. We have the traditional maps, the humanized maps or modified engineered maps, and several other formats that will be discussed later. Another key point is the safety of these products and the different approach for developed product that is structurally stable and with favorable safety profiles. We have a set of knowledge that allow us to modify the aggregation profile, the structural stability if needed. Even the monoclonal antibodies are quite stable. We know today how to modify that in order to do it even more stable, how to modify the immunogenicity, how to modify the properties for obtaining a good PK/PD profile, or how to do them more stable, easy to formulate, with good properties for resistance for freeze-thaw cycles. And no less important, we have the experimental analytical tools to evaluate that in the lab before entering in the clinical trials or even preclinical trials. We have a set of experimental tools to predict how the antibody will be performing in vivo, and that is very, very important for the success of these products. We can design an antibody that will be successful even before we start injecting one patient. And well, we have also commercial and strategic drivers that are based in strong intellectual property opportunities. All of these products that were shown before are protected by IP, by patents that ensure that the companies will recover all the investment in the development. The rates of success are really high, and we have a market, of course, for personalized therapies or for diseases that are really extended in the population. And for example, as Leda was presenting before, for adalimumab that has several indications, it's not only one indication, several indications, we have that the accumulated sales for the products were $200 billion. It's a lot of money for AbbVie. Well, of course, they had a patent that protected the antibody, and when the patent was near to expire, they create a variant that it was based in a difference in formulation that extended the period for them to obtain a revenue of this antibody. Well, taking a little bit more attention in the manufacturing of these antibodies, we can say that, well, from the introduction of the CHO cells, we had the start of industrial production. Then after that, we have a wave that was focused in improving the bioprocessing through media formulation, nutrient feeds, and bioreactor control that increased the original titers that were below 100 milligrams per liter to 1 gram per liter. In that time, the standardized protein A chromatography also simplified the process of purifying the antibodies. In the early 2000s, we had a breakthrough for cell line engineering. We could obtain better cell lines, and the titers reached 2 to 3 grams per liter. And we started there hearing about some companies that scale up the process to 20,000 liters bioreactors. In the mid of 2000, well, the platform concept was dominant. We have a very clear way to produce monoclonal antibodies, and companies explore some variants, for example, high-capacity resins, improved versions of protein A with more resistance to cleaning agents. For example, membrane chromatography was also implemented in large scale. Well, after that, the high titer era pushed the titers to beyond 5 grams per liter, and the emphasis was in reducing the cost, because it appeared the problem for health payers for acquiring or paying for these products. Therefore, we needed cheaper antibodies, and the competence also was harder at that time. And well, after the 2010 and today, we have the introduction of new paradigms like quality by design or process analytical technology. The criteria or the idea of these technologies is to provide more flexibility in the manufacturing. Because every change in this industry is very expensive to be implemented, and therefore these ideas of quality by design and process analytical technology should provide more flexibility for the supplier of monoclonal antibodies to make changes if needed without affecting the quality of the product. Well, this is a typical upstream processing. The process starts in the cell bank. With the cell bank, we take a vial from the working cell bank, and we start the expansion during several days. This typically takes around 20 days, this step. And then we start, when we finish the expansion in the inoculum phase, we see the production bioreactor with a typical size today for the fed batch modality of 2,000 liters, 10,000 liters, or 15,000 liters. That stage in fed batch takes around 14 days. And after that, the cells are separated, and we have, at the end, the harvest free of cells in a vessel ready to follow the downstream processing. Regarding the modality of the culture, we have, of course, now these are the two dominant modalities, fed batch or continuous processing. Fed batch processes are not common nowadays. It were replaced by fed batch processes. And well, in every case, the monitoring of the culture, the optimization of the conditions is the key for reaching high product titers with a low cost. Essentially, the media is a major contributor of the cost of this stage. In the downstream processing, we have several steps. Most of them start with protein A affinity chromatography. After dilution of the chromatography, the material is maintained in a vessel where the pH is adjusted to 3.5 in order to inactivate viruses, potential viruses that could be present. Then that material is filtered, and a conditioning of the material is performed to go through the second chromatography step that is a polishing step, and usually as a third chromatography step that is used also for polishing purposes. Finally, the viral filtration with a nanofilter, and the final concentration and filling to obtain the drug substance there in a concentration that is usually 150 to 100 milligrams milliliter. Well, this is the typical platform of 3 columns, a protein A and 2 cation and anion exchange chromatography. Well, the order it could change. In some cases, the an additional step is added, or the mixed mode where hydrophobic chromatography and cation or anion exchange chromatography are combined in a mixed mode chromatography. A different platform that is more interesting because of the cost of processing of only 2 steps, where protein A is the first chromatographic step, and the second one is a weak partition and mode, where the sample is the sample to be loaded in the chromatography is modified or conditioned in order to have a very weak binding of the antibody to achieve a very early elution of the antibody, whereas the impurities are bind to the chromatography and elute later. These are the typical columns for the industrial scale, and we have several impurities that must be removed during downstream processing. Host cell proteins and host cell DNA are typically removed in protein A step and also in the polishing steps. We have some leachables from the protein A, I mean small parts of protein A that elute with the product that are removed in the polishing steps, and endotoxins and viruses that are adventitious contaminants are removed in the low pH inactivation or viral filtration or nanofiltration, that is essentially the same, whereas endotoxins could be removed in the polishing steps. And aggregates and fragments are really the most difficult things to be eliminated, are removed during the polishing steps. Aggregates can be the most difficult things to be eliminated, and also fragments or related proteins that are very similar to the main product. And well, of course, other process-related impurities, such as media components that must to be removed, and typically they are removed during protein A and polishing steps. Regarding vaccines, well, we have a history of 200 years of vaccines that had been used globally for immunizing population. We have the live vaccines that are not used, not using the modern platforms, but we have some attenuated vaccines or non-live vaccines where the organism is killed, or we use some parts of the organism to elicit the immune response. Newer platforms are protein polysaccharides conjugates or viral vector vaccines or nucleic acid vaccines that appears more recently. As Leda was explaining, we have a biopharmaceutical life cycle that is very long. We start with the discovery, the process and analytical development, the clinical manufacturing, the regulatory review and licensure, the commercial scale-up, the distribution and logistics, and the post-marketing and life cycle of the product. For the discovery and preclinical, we have different components, starting with antigen selection and design, small-scale experiments, preclinical testing. During the process and analytical development, we have to select the technology to produce the biopharmaceutical a vaccine or biotherapeutic with that is a lot of work here that typically is expected to be done in a very, very short time. And the clinical manufacturing implies technology transfer from the lab to the GMP facility. That can be a little bit a difficult task. But from that GMP material, we can run the first clinical trials. Some work must be done there to adapt the process to a GMP environment. And with that material, we can run the first stability studies according to the specific guidelines for stability studies. Here, after the first results, we can continue the optimization. However, any change from this phase will result probably in a delay of the project. Therefore, this stage is very critical for the project. After that, the regulatory review. It is also a very tough stage. It will depend on the feedback of the authorities. It's very important here to have very keen people in order to have the right people writing the documentation according to the expectation of the authorities that will review the information, all the information of the process, of the product, of the development, etc. The commercial scale up, it can be done before or after the approval, typically after the approval. In that stage, additional validation work must be done. The distribution and logistics is very important, especially for vaccines, where the cold chain management can be very important to reach different populations, different regions in the planet. And of course, the post-marketing surveillance and the obtention of real-world data that is very important and probably will trigger the need of decreasing or reducing the cost of goods in order to have more competitive prices for this new product. Taking into account those steps that take a lot of time, we have the explanation or the main explanation of why new modalities emerge. We have traditional platforms as the platform that I explained for the production of monoclonal antibodies that have key limitations. For example, for vaccines, we have a long development cycle. Many of them are cell-based. The production is cell-based. Therefore, it takes a lot of time. I mentioned a 50-day batch that is quite normal for cell-based products, and a slow response when new pathogens emerge. Therefore, this is a constraint, especially for vaccines. The design of antigens is also constrained by technological limitation of growth or purifications. And in the case of monoclonal antibodies, we have a high cost. In fact, we are seeing lower cost today, but they are even high cost, and those products are really, really expensive. Mammalian systems take a long time, and we can see some aggregation issues or immunogenicity issues. Even the efforts to design a proper molecule to avoid aggregation or immunogenicity issues, we can see that in some products. For the emerging modalities, we have some platforms that allow a rapid design of the product, and a cell-free platform that is more scalable. Sorry. And we have some other modalities with enhanced specificity. We have the design of a molecule that is very interesting because we can add many antigens in the same product. And that opens the door to the access to more targeted products for personalized medicines, especially for those diseases where we have a low number of patients and we need a small amount of product for attending that need. Therefore, the new modalities bridge gaps in speed, precision, and scalability. Those are the gaps that are attacked with the new modalities. However, these new modalities, we can say that new modalities didn't appear suddenly, are based in decades of science. For example, for mRNA vaccines, we had the first results that were published in the 90s. We have the results not only about the efficiency of the liver naked RNA in an animal, but also the technology to stabilize that molecules. The core concept was clear even in the 90s, but we had some problems to address that were instability of the product, immunogenicity, especially because of some components of the liposome, and the inefficient delivery. The advantages of these platforms, well, is that the technology is very safe. We don't have an integration in the host, in the patient genome. The efficacy, because the sequence can be very well designed and engineered, and the manufacturing and scalability, because we don't need any cell, and therefore, well, the process is quite simple. And the platform is also very, allows coding multiple antigens in a single vaccine. How we produce the vaccines with this technology? Well, we have a plasmid template. Then we produce the mRNA in vitro, I mean, without any cell participating. They are only enzymes. And then we produce the nanolipids that will protect the mRNA from the degradation. Therefore, we have an upstream production and downstream production where we remove the enzymes and other regions that were used during upstream, and then the formulation, the three-step processes, where a single reaction in a 5-liter bioreactor can produce 1 million doses of a vaccine. This is a different explanation, but here we have the DNA template and the enzymes that contributes to the production of the mRNA, then the downstream, where the impurities are removed, and finally the formulation for encapsulating the mRNA. We will see for sure this in more detail in the specific talk of this topic. And of course, the global pandemic validate the technology. In less than 1 year, we had 2 vaccines that were approved, developed, and authorized, and we have a very high efficacy. where clinical trials demonstrated exceptional higher efficacy for the vaccines producing using this technology. We have more and more products that are being developed using this technology. We are beyond COVID. We have several other targets. Different companies that were the pioneer in this technology are publishing results of taking into account different diseases with different areas, in different areas, even cancer vaccines. And also it is, well, development of new technology to address some limitations of these technologies, like the supply chain of the vaccine, because of the cold chain that is a constraint when we need to deliver the vaccine in some regions of the world. Some work is being done for improving potency and duration of the response, and new carriers are being introduced in order to obtain vaccines that are more that have more efficacy. Well, some things that are being investigated yet, for example, the duration of the antibody response is something that must be tested in long clinical trials studies. The vaccination of a special population that is already being investigating, the safety and reactogenicity when we talk on some components of the vaccines. We know that 2 to 5 individuals per million can have a reaction that is an anaphylactic reaction because of the PEG that is a component of the vaccine. 40% of the population has antibodies, pre-exciting antibodies for PEG, polyethylene glycol. And as that component is present in the vaccine, some individuals have that anaphylactic reaction, and that is something that's being studied nowadays. And well, also the global access and acceptance of the vaccine is something that is a point to work. For example, taking into account the acceptance point, very recently the US Minister of Health announced that they will down some economical support for mRNA vaccines. The justification is that they are not the efficacy is not enough for certain disease, especially for respiratory disease, and well, that could have an impact in the development of new vaccines. I mean, that is something important that also is a political matter too, but is very important, mainly taking into account that from United States, most of the money is pushing or not the development of new vaccines. We also have more interesting things. For example, the World Health Organization has organized a special line where they encourage the adoption of mRNA vaccine in certain countries. They starting in Africa. The goal is to strengthen the regional production of this kind of vaccines. They have some key achievements that were already finalize it. The technology transfer that was completed from a company that is Biovax to two different sites in Africa. That stage was completed, and they are planning now to build a commercial scale GMP production of vaccines in South Africa. And well, it's an interesting program, I think, because well, it wants to put this technology in more areas in the world instead of putting all the knowledge in certain countries or certain companies. Another interesting thing that I would like to comment before ending my talk is this breakthrough therapy that was achieved very recently. Maybe you already heard about that in Huntington patients. Huntington is a disease that is, well, it's a terrible disease that affects motor systems, well, cognitive functions. Well, very recently, very good news arrived after using gene therapy, where a virus that is not a replicant, not infectious, deliver in the brain, in the neurons, a specific DNA fragment that allows the neurons to produce a microRNA that allows, after the infusion of this product in the brain with a specific surgery, allow to lower the amount of a toxic protein that is the cause of the disease. And well, in fact, the results were highly positive, with positive results in more than 70% of the patients. Only a few patients in the world have this disease, but the disease is really very, very damaging for motor and for cognitive functions. And well, this is something, a breakthrough therapy was approved, and it is also a great news for science and for patients and for their families. Well, takeaway messages. Well, we have the monoclonal antibody era, where a platform is the strength of the production of the manufacturing, of these antibodies. I mean, the platform for the production or manufacturing is the strength of these products, and many other characteristics that are from the products that are high clinical utility, commercial success. We have regarding manufacturing, we have a process standardization based in the introduction of unique cells that are CHO cells that are very robust, and proteinate chromatography that where the purity of the product is very predictable with a very good recovery. We have a yield revolution where we have titers beyond 5 grams per liter, even 10 or 20, according to the bibliography. And next gen manufacturing that is based in the introduction of certain concepts to allow more flexibility during manufacturing, that allows reducing costs and making the manufacturing more simple. Well, also the continuous bioprocessing, that is something, as Aleda was presenting before, that is quite old, but nowadays companies are taking more advantage from continuous bioprocessing in order to reduce the cost of antibodies manufacturing. Well, we need new modalities and innovation in order to have new products faster, with more precision, and with manufacturing platforms that are more scalable. We have for that different formats that will be discussed in this course with specific talks. And, but the idea is the foundational science for all these platforms are not new. Maybe the COVID pandemic had a positive, at the end, a positive result that was pushing these technologies and demonstrated that, well, they are safe. Really, they are safe and efficacy, and had efficacy. The new technologies can be a breakthrough in the speed and versatility of those platforms with multiple manufacturing advantages, producing these therapeutics without any cells, and therefore pushing for more speed in the development and the production cycles. They are really effective. Yeah, we have many doses of a vaccine, for example, in a small scale that is very attractive. And the efficacy is also a good point for these new modalities. And also the safety. Whereas we have some problems to be addressed, a good progress is being made about that. The future is the expansion of the pipeline with many other diseases to be targeted by these new modalities. A second generation engineering to eliminate the cold chain, even with the antibodies, to trying to push for product with new formats, conjugating the antibodies with drugs or bispecific antibodies. I mean, the engineering of the products is the future, both for these new platforms or for mature platforms. And of course, the importance of obtaining products that are more affordable and that can be produced in several regions, not only in some countries. And well, that is all my messages, and I hope you can enjoy some new things. And of course, if you have any question, if I can do it, I will respond it.
So we have a lot of time for questions. So does anyone have questions?
Thank you for your presentation. And I was wondering, you said for MAB, there are many points which becomes easier to produce it, and like the scalability and everything. I was wondering if soon the mRNA vaccines also will be the same, like easy points on the manufacturing, if that's a point in which like the technology can evaluate or move up to, or if it's a singular point to the MAB vaccines. I don't know.
Well, I understand that for MABs, we have a high installed capacity. We know already what are the weak points. We have an accumulation of a lot of knowledge, and especially that high installed capacity. I think that it is something important. decreasing the cost of producing it, I think that they will be supplemented by new technologies. I don't expect that new technologies replace the production of maps, for example, but I understand that will supplement for new diseases or for personalized medicines, for example. Regarding these new technologies, I think that according to the knowledge is accumulating in manufacturing processes, more new things will appear, probably new concerns as used to be. But, well, of course, if the business pays all the new developments, more capacity will be installed also in different regions to overcome those difficulties that will arise. I think that when you know more about a technology, you realize that some risk appear there, and you need to work to mitigate those risks. I think that, well, it's a newer technology. It will supplement the products, the map products, and we will have more options for treating diseases that nowadays have no cure.
On top of that, do you think that if we have like other approaches to mitigate those diseases, the cost of mAb vaccines and treatments will drop? Because nowadays it's really high and
really high, yeah. Yeah, I expect, well, I think that in this course, people that is more expert than me will talk about that. I think that new work is being done in technology for purifying maps or producing maps, new cell culture media. The competence in cell culture media is higher nowadays. Therefore, I expect that the cost will decrease. The biosimilars, for example, demonstrate that prices can be lower because the impact in prices used to be very, very fast after the appearance of a new product in the market. Therefore, I think that there is a space to decrease the prices. I think it's a challenge for companies that are already producing maps to change their processes in order to reduce costs. I think that there is a lot of space for that, but it's challenging, of course.
So, first of all, thanks for your presentation. You said on the safety and development slide that it's possible to modify the monoclonal antibody to be more effective. So I would like to ask you how does modify works, if it is something that you change on the final product or something that you change in the cell line that will produce the monoclonal antibody?
Well, engineering of antibodies allows to separate variants of antibodies that could be dangerous if they are present in the product, or you can modify the interaction of the Fc part with effector cells in order to have more efficacy and needing lower doses of the antibody. For example, through engineering of the molecule, you can facilitate the downstream, you can obtain more efficacy in the response, and therefore lowering the doses of the antibodies that at the end results in a better safety profile.
More questions?
And hi, thank you for the presentation. I have a question regarding mRNA vaccines. You showed us that we can develop antibodies, for example, for PEG through vaccine excipients. So my question would be, what what are the risks of developing these antibodies against PEG or other excipients in multiple vaccine doses, like COVID vaccines that we have to take like 2, 3, 4 doses? And how can a person know that they developed this? And if they don't know it, if all vaccines that use the same delivery technology, like lipid or PEG, would stop working for them?
Yeah. Well, I understand that many of us already have antibodies against PEG, pre-exciting antibodies. I don't understand that putting different doses of vaccines using this technology will increase per se the appearance of those antibodies. However, the risk of those reactions, type anaphylactic reactions, is there, and therefore new formulations for the vaccines must to be developed. However, I understand that the risk of having such anaphylactic reactions is very, very low. However, it exists, and it drives the development of new formulations. I don't expect, according to my knowledge, that using more and more this technology will increase significantly the appearance of those anaphylactic reactions. It's more something that an individual has a specific sensitivity to this reaction, but most of us already have these antibodies. Okay?
More questions? So I have one. Regarding gene and cell therapies, we know that we have regulatory issues and also the costs. So do you think this will be the future for personalized medicines or in treatment?
Yeah, well, the cost is a very, very important thing. As these therapies many times are injected only once in life. I mean, you don't need repeated injections. You only have one injection, for example, for Huntington disease and some others. The reality is that the price is really, really high. Of course, it's the difference between life and death in some cases, and well, the price, it's something that is related with that. I understand that the prices will decrease. However, in the meantime, I think that it's not easy to use in any context. Even for United States or Europe, a lot of controversy is already regarding the price of these treatments. That is really high. For $500,000, for example, for a treatment, it's only one injection, but well. It's very, 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 very costly. Yeah.
I have one question about cell lines, because if you see the products that are already produced in mammalian cells, 80% or more are produced in CHO cells. So what do you think that impairs the growth of the use of other cell lines? Is that so many difficult to have a validate process with other cell lines, or CHO is really unsuperable?
Well, I think that CHO cells are very well known, are safe, and those safety things are very important, and also the development of new ways to generate these recombinant cell lines that allow to have products in a tighter between 5 and 10 or even 20 grams per liter. I think that make them very, very interesting platforms. I mean, even if you don't have by yourself the knowledge to do it, there is a number of companies that can produce a cell line of such characteristics, maybe by $1 million or something like that. But well, it's quite in maybe 6 months, for example, or even less. I think it's a matter of speed. if you have an idea, a good product, a good antibody, you don't want to expend time developing a new cell line. That is the reason, I think, because of the CHO cells are very robust and are very, extended. Well, if you have a higher titer, for example, you will have a bottleneck in the downstream part, and maybe you will have a different problem. That is my understanding about the situation.
So one final question.
Actually, I just wanted to comment your question because when you were talking about the cost of, I can speak only about gene therapy based on AAV, which is the most widely used viral vector, but a lot of it is about the process, whether you have like adherent cells or suspension cells, and how you do the scale up. So if you're talking about adherent cell process, it's difficult to scale up until a limit where you have to scale out. So you have to do multiple things. So if you have suspension cells, it's easier to scale up. And another point is if it's with transfection, because transfection is the most expensive costs of one of the most expensive costs. So one alternative is to use stable cell lines like we do for monoclonal antibodies. So there's been a lot of work developing stable cell lines for AAV production, because this way you reduce the costs, you don't use the transfection reagents, and it's easier to scale up. So depending also, like some products, you require like a small dose and some large dose. For at least for the one where you need large dose, you can produce like, I don't know, scale up to 2,000 liters, 20,000 liters. You cannot do this with the adherent or transfection process.
Only a short comment. Yeah, I think that the price is not only something that is related with the manufacturing process, where probably, well, more improvement must be done, but also it's related with the number of patients. If you have a very, very little number of patients, well, the price it used to be high, especially in these years where the validation of the technology is already happening for gene therapy, for example. Therefore, the small number of patients are paying for the development of that product, not only the manufacturing. Exactly. Exactly, yeah.
So actually we have time for one more question. Does anyone have?
Nice to see you.
Nice to see you too.
I'd like to know all your presentation, specifically about Latin America, or what do you think we can do different than US or Europe? I mean, you have your experience in Santa Fe, and do you think we can compete side by side, or can you comment, please?
Well, I think that many things could be improved in order to have a process that are, I mean, more with lower cost of production. However, it is difficult today to think a new development thinking only in our market. I mean, thinking a new development only taking into account our markets is difficult because of the size, because you have the competence of big companies that will enter too, and will put a pressure on the regulatory agencies. I mean, at the end, it's very important to try to be innovative, to have lower costs in the process, but you need to think in a global market in order to have a business that is consistent. Therefore, you will have less opportunities to innovate. I mean that innovations
People are
to be validated by others in order to be, to get the approval in those markets. I mean that I think that the only way to try to be successful in our countries is to align our development and our manufacturing capabilities with the worldwide market. In my opinion, is the only way to do it nowadays, at least.
Guilhermina, hello. Good talk. You said about biological production, and we are in a university, a local for produce knowledge, basic science, and I see you work with two words, university and industry. How you see this technology transfer to university for industry in South America, in our countries? How to see to increase this movement, or is difficult to cross these values?
Yeah, no, I think that this the key point is the integration between university and manufacturing sector, because some developments from the university sometimes are difficult to implement in manufacturing, especially taking into account that you, in general, you need to be aligned with the manufacturing technologies that are, international rules. And therefore, the only way that the university produces knowledge that can be adopted by the company is if they know exactly what the companies are needing. Because if you have a researcher that is very good but doesn't have the end of his work in mind, probably they will spend a lot of time in things that probably at the end will not be able to be transferred. I mean, it's very important that researchers go to the companies, understand the needs. That is more probably that the companies come to the researchers. That's why I am telling that. And understand the needs, understand the problems, and understand how a new product can be at the end a business in order to be incorporated.
So thank you, Guilhermina, for your wonderful talk.
Thank you.
So right now we have a 15-minute break. We have toilets on Block G, and the coffee break will be here. So feel free to enjoy yourselves and have a break.
People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacterial light chains and 2 heavy chains. The solution to this problem was a Nobel Prize-winning People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes
Hello?
Pode. Pessoal, só falando que vai voltar aqui, tá, a sessão? So, hello again. So we're going to move forward with next presentation from Patricia. Just a quick announcement. Since our coffee break was a little bit extended, our time for lunch is going to be midday 45 instead of 12:30. So, and we're still coming back at 2 p.m. So, just so you know. To introduce Patricia, just a second. So, Patricia holds a PhD in cellular and molecular biology from Fiocruz, with part of her research conducted at the AIDS Vaccine Research Lab, AIDS Vaccine Research Lab in Madison, United States. She is currently leading the implementation of the first RNA development and production hub in Latin America at Biomanguinhos, Fiocruz, focusing on fully Brazilian-developed RNA vaccines and therapies aimed at strengthening regional self-sufficiency and expanding RNA technology access in Latin America and Africa. So welcome, Patricia.
Thank you, Renata. Thank you, Dr. Leda, and all the organizers for inviting me to be here today. And I will speak a little bit about mRNA-based therapeutic and vaccines, and also about our experience in Fiocruz implementing this platform during the last 4 years. So I'm Dr. Guilhermina already talked a little bit about that. So I always start my talks showing this seminal paper published in 1990, showing that RNA could be a vehicle for foreign protein expression in vivo, in muscle cells. So it's important to know that this platform have not appeared during the pandemics. It has been developed during the last 30 years. So there were a lot of hurdles that needed to be solved in order for us today to have this platform being used as this is being used now. So at that time, there were a lot of problems related to the use of RNA as a vehicle for new drugs development and vaccines. So RNA is high immunogenicity. Foreign RNA is recognized as a foreign molecule, so it elicits immune responses against, because if you think viruses are foreign RNAs invading the cells. So cells have a lot of different sensors to answer to this threat. So RNA per se, the natural RNA is a molecule that elicits immune responses. The low stability of RNA, of course, this is a very unstable molecule, and high production costs at that time, during the beginning of the 90s. So at that time, the whole attention went to the DNA platform. But during this time, it was difficult to move forward with DNA vaccines and products because DNA has a lot of other problems related to the molecule, such as for the DNA, you should transfect the cell, and the DNA need to reach the nucleus to be translated. So there were a lot of problems with this platform during the last 30 years. So there were some researchers that kept working on RNA and improving this platform. Before we go to the how the researchers solved these problems related to the RNA platform, let's talk a little bit about the technology. So the most known technology related to the RNA products is the non-amplifying RNA, the one that is used in Moderna's and Pfizer's vaccine, the traditional one. The molecule is very similar to our own RNA. I will talk a little bit about this structure later. We have also the self-amplifying RNA, where we have the gene of interest, but also a replicase coming from a virus, in general, an alpha virus. And we have a process that I will talk in the next slide of self-amplifying this gene of interest. We have now also the trans-amplifying platform, where we put this alpha virus replicase in one molecule, one RNA, and the antigen in another, and we co-transfect these two RNAs to obtain the molecule of interest. And now we have also the non-amplifying circRNA, where we use an iris element without, and we have translation without cap in the cap dependence. So these two types of RNA are more recent. We are still learning on that, but this technology is as old as non-amplifying RNA. So this is how the technologies work. So this is the conventional RNA, the non-replicating with the cap, the traditional elements of RNA, the cap element, the poly-A tail, the 3-line, 5-line UTRs, and the coding, the gene of interest coding part of the RNA. And this is the self-amplifying with the addition of the replicase. And for the conventional one, when it's delivered to the cell, the RNA will be delivered to the cytoplasm, and it will be translated in the protein of interest. And in the case of the self-amplifying RNA, it will be delivered to the cell, and the first gene to be translated will be the genes that encodes the replicase. So the replicase will amplify this coding gene region. And with this amplification, the gene will be translated, and then the coding gene will perform the role that we wait for this molecule. So coming back to of course, this is much more developed today than this technology, but this self-amplifying RNA technology is being considered the next generation of vaccines based RNA, because of the lower dose necessary, we have mechanisms here that will improve the immune responses. So maybe we will have better memory formation with this technology and lower costs of production. So coming back to the evolution of this technology, during the last 30 years, there were a lot of research being made with this molecule in order to have the technology we have today. So these elements that we know that are part of now we are going to talk only about the non-amplifying RNA. So these elements, the non-translated 5-line, 3-line, the capping element, and the size of poly-A tail are very important to the RNA technology. And these regulatory elements were very well studied to improve the protein expression and stability, and also modifications in coding sequence. The first major modification that we perform in this coding sequence is the use of nucleotide modified in this RNA production, and also coding optimization, and all of these modifications will improve, will diminish the recognition of the RNA by the sensors, cell sensors. It will improve the stability of the molecule, the protein expression, and making this RNA durable with increased lifetime and higher translability. So as I told you, the first great major modification that we were discovered to stabilize the RNA was to use modified nucleotides. So there are a lot of different nucleotides that we can use to substitute the regular nucleotides in these RNA molecules, but the most important one that's being used in the vaccines that we use today is N1-methylpseudoridine. N1-methylpseudoridine was described by the wonderful work made by Professor Katalin Karikó and Drew Weissman, and because of these discoveries, they were awarded with the Nobel Prize. What happens is that when we transfect the cells with the regular RNA, with the regular uridine, this RNA is sensed as a foreign molecule. So the RNA will elicit different types of pattern recognition receptors in cells, and this recognition will elicit the innate immune responses that will, the release of type 1 interferons will increase the capacity of the cells to eliminate this RNA. So what happens is when you transfect the cells with your regular foreign RNA, it will be recognized as an strange molecule, and it will be destroyed by the cell before it's able to produce your protein of interest. When you use the N1-methylpseudoridine replacing the regular uridine, this recognition is impaired by this methyl in uridine. So what happens is that this RNA will not be recognized as a foreign molecule, and it will, of course, be translated and have this protein of interest being expressed by the cell. So this is the first great discovery related to this RNA platform. But when we talk about the work that have been done by Katalin Karikó and Drew Weissman, they were trying to use the RNA to differentiate iPSCs. And the RNA platform was mainly developed using therapeutic area as a model. But we are talking here about preventive vaccines. And when we talk about preventive vaccines, maybe a little bit of inflammation would be good to elicit the immune responses and increase the memory formation. And during the development of our candidate in Fiocruz, we were facing a problem because N1-methylpseudoridine is patented by Moderna. So we could not use N1-methylpseudoridine in our formulations. So we started thinking about using the pseudoridine. So I talked already about the unmodified uridine, the regular uridine, when we have the highest interferon and cytokine expression and inflammation, and the lowest expression of the protein of the interest. And on the other side, we have N1-methylpseudoridine, where we have the opposite. The lower inflammation elicitation, but the highest protein expression. In the middle of this way, we have pseudoridine, where we have a little bit of inflammation, but also we have enough transgene expression. So we started thinking about using pseudoridine to see what would happen, because we were not able to use N1-methylpseudoridine. So we've performed a single dose assay with our candidate, immunizing mice with our vaccine with one dose, and following the animals by a lot of different days. We were sacrificing 3 mice per day and collecting material. And what we saw is that when we use pseudoridine instead of N1-methylpseudoridine, we have higher inflammation, higher production of chemokines, and also different genes expression in these mice, in blood of these mice. Related to the interferon pathway, there was no difference between pseudoridine and N1-methylpseudoridine, which is good, because it means that the pseudoridine RNA is being recognized as the same way the N1-methylpseudoridine is being recognized as foreign RNA. And when we look to the plasma cytokines production, we were able to see that pseudoridine is really slightly more inflammatory than N1-methylpseudoridine, and maybe it could be good for our memory formation and vaccine efficacy. And what we saw is that when we immunize mice with pseudoridine, we were able to see much more interferon gamma being produced by CD4 and CD8 cells. And also we could see that at least for CD8, the memory formation is better than when we use N1-methylpseudoridine. So we are preparing this paper to submit, and we are investigating better the use of pseudoridine instead of N1-methylpseudoridine when we talk about preventive vaccines. When we look to the immunogenicity and the efficacy comparing pseudoridine and N1-methylpseudoridine, we immunized mice twice with both RNAs, and we collected samples for immunogenicity 14 days after the second dose, and part of the animals were challenged with SARS-CoV-2, and we collected also the material for these mice to see the viral loads and evaluated the survival. And when we see here that it's better when we use pseudoridine, the total antibodies responses is higher than using N1-methylpseudoridine. It was not possible to see difference when we talk about the neutralizing antibodies, and also when we talk about T-cell responses. But when we look to the viral loads in oropharynx and lungs, it was possible to see a difference when we immunized mice with pseudoridine and when we immunized mice with N1-methylpseudoridine. And there is no difference in survival. So we hypothesize that using pseudoridine, we will get better immune responses and memory formation. So talking a little bit about the production of the RNA, I think that Professor Guilhermina already talked here today. So we use for the RNA production linear DNA template. So we obtain the DNA using the regular process, expanding E. coli. We have in our plasmid the T7 promoter, the protein coding sequence, and the poly-A tail sequence. And this linear DNA template is transcribed in our RNA molecule. This molecule is purified. There are a lot of different types of purification, using columns, using beads, and for the main impurity of this process is double-stranded RNA. So this is a figure of the whole process. So we transfect the E. coli with our template, plasmid template. We cultivate the E. coli. This process is being developed at Biomanguinhos by recombinant laboratory team. This E. coli is cultivated, is filtrated. We perform the lysis of these cells. We purify the plasmid, linearize. This linearized DNA is used as a template to produce the RNA. The RNA is purified and goes to the encapsulation that I will talk a little bit later. And the second major discovery related to the RNA platform was the development of delivery systems. So the naked RNA is very unstable, and in our body there are a lot of RNases. So injecting naked RNA in mammalian body is not a good idea. It will be destroyed in a few minutes. So people that were working on this platform started developing a lot of different kinds of delivery systems. So the most very well-known delivery system is based on lipids. I'll talk a little bit about the composition of these lipid nanoparticles later, but there are a lot of other kinds of delivery systems that could be used, like polymer-based delivery, peptide-based delivery, virus-like particle delivery, cationic nanoemulsions. Naked RNA is not so good, but also electroporation. But these delivery systems are able to protect the RNA from the hydrolytic enzymes and also from RNases. So we know today that there are a lot of roles of these delivery systems. It's not only to deliver the RNA, it elicits, so it's part of the immune responses. It will elicit also the immune responses. So I brought here some of our examples of that. So this is comparison between a polymeric and the lipid nanoparticles for the delivery of self-amplifying RNA vaccines, because one of the challenges for the development of self-amplifying RNA is the delivery, because it's a longer molecule compared to the conventional one. So for example, self-amplifying RNA has more or less 11 kb compared to the spike conventional RNA, which is 3 kb. So it's longer, and it's more difficult to encapsulate. So here they compared two different delivery systems for this self-amplifying RNA. A polymeric one called PA ball, and the traditional one using lipid nanoparticles. So these are different composites. So I will talk a little bit about the composition of the lipid nanoparticles, but in general, we have 4 types of lipids in these lipid nanoparticles, and in these different lipid nanoparticles, they changed one of them. And they were comparing these different compositions. And this is the PA ball. And this is a very traditional assay to see how much RNA is going inside the cell and is expressing your gene of interest. So we put luciferase as a gene of interest, and we inject the animals with luciferin, and it will appear in this equipment, and with this luminescence we measure how many RNA is going inside the cell. And as you can see here, PA ball is much better to deliver this RNA to the cells than the lipid nanoparticles when we compare. So much more luciferase being expressed when you use PA ball than when we use the different types of lipid nanoparticles. But when we talk about the immunogenicity of the vaccine, this is influenza vaccine as a model, and we look to the PA ball RNA delivery. We can see that this nanoparticle is much better in eliciting the immune response than the PA ball. So it's not only the delivery, it's the role of the nanoparticle, and the composition of the nanoparticle will play a very important role in this immunogenicity of the vaccine. So here is the body weight after the challenge, and as we can see here, the PA ball delivered very well, but it's not protecting the animals. So it's very important to pay attention to the delivery you are using for your vaccine. So as I told you, the lipid nanoparticles are the most very well known and used delivery system for the RNA platform. This is composed by, in general, 4 types of the classical one at least, but now we are seeing the development of other kinds of lipid nanoparticles, but the classic one that we are using in Fiocruz and also others are using all over the world is composed by 4 elements, 4 types of lipids. The ionizable cationic lipid, that I will talk a little bit about that in the next slides. The helper lipid, cholesterol to give a structure to this lipid nanoparticle, and the pegylated lipid to improve the half-life of this lipid nanoparticle when it is injected in the organism. And when we talk about the process of encapsulation, we use microfluidics, where we put the nucleic acid. This goes to the aqueous phase, and lipids in organic phase, normally in ethanol. And we use we improve this process, testing different ratios between these two reagents, and also the velocity of the encapsulation, the flow rate, a lot of different parameters that will enable us to mount the proper lipid nanoparticle and to achieve these purposes of this delivery system. So it's very important to know the LNP components, as I told you already, and I will talk you about the role of ionizable lipids in the next slides. The in vivo stability, the structure and morphology of these lipid nanoparticles, to homogenize the size, the dispersity, and achieve the storage stability. And we have these parameters related to the endosomal escape that are very important in this case. So one of the most important components of this lipid nanoparticle is the ionizable lipid, because the ionizable lipid will allow this RNA to enter the cytoplasm. So the ionizable lipid is a component that is cationic in lower pHs and is neutral in neutral pHs. So when we encapsulate when we are performing this encapsulation, this nucleic acids buffer should be at lower pHs, like 4, 4.5, because in this case, this ionizable lipid will be positively charged, and it will kept the RNA, which is negatively charged, to it will capture the RNA, and the lipid nanoparticle will be formed. And when we inject these lipid nanoparticles, these are two types of ionizable lipids. This one was the one that's used in ONPATTRO, silenced RNA product, and this one is another one that's being tested to deliver the RNA. So when we put these ionizable lipids in this composition, it will be it will suffer the endocytosis by the cell, or macropinocytosis, and when this endosome will lower the pH, it will make this membrane, this lipid nanoparticle membrane will fuse to the cell membrane, and this RNA will be delivered to the cytoplasm. So as you can see, the role of, and this is because of when this endosome is going inside the cell, the pH is lowering. And because of this lower pH, the membranes will fuse, like viruses. It's like the viruses mechanism. So it's very important to use a very good ionizable lipid in order to have a very good vaccine or product. So this is an example of what I'm saying. This is a comparison between this SM102 is the Moderna ionizable lipid, ALK 0315 is the Pfizer's vaccine lipid, and this is one that this article was testing. And when we compare the same assay that I showed you for PA ball, it's possible to see that SM102 is better in delivering the RNA than the other two lipids, ionizable lipids. And during our development, we had also to test our own lipids. because it was not possible to use OAK or SM102 because this was patented by each one of the companies. So we started looking for a good lipid that we could use freedom to operate. And these are the results of some of our tests. So here the control is the SM102. And this is 3 different ionizable lipids that we've been testing during this period. They perform for T-cell responses. So we can see that formulation BI was very good in eliciting T-cell responses. And when we are going to see the total spike anti-IgG, it's also possible to see that formulation BI was the best one. And also when we talk about neutralizing antibodies, this formulation BI was the best one. When we are going to see the viral loads, it was possible to see that formulation BI was also the best one in these tests. But even with these different immunogenicity profiles, all of these formulations were able to protect against the lethal challenge with SARS-CoV-2. But of course that we moved forward with the best one with our candidate. This is this biodistribution assay to show that the RNA is expressed during a period of time in the animals, as you can see here, and by 336 hours it will be gone. So this is a kind of result that we need to show to ANVISA to prove that the RNA will be eliminated during this period. And in terms of RNA industrial production, I talked a little bit also. We have the production of plasmid in GMP. Plasmidial DNA is an intermediate product that will be used as a template to transcribe the RNA. And this RNA will be encapsulated and formulated and go to the fill and finish, sterile fill and finish. And I think that you already know, but it's important to say that this is except for the pDNA production. Here the process is enzymatic, so much simpler. The footprint of the facility is very small. And this is the main platform that are being considered as ideal for answering new threats, pandemic threats. And of course, I don't have time to go through each one of these tests, but I wanted to show you all the tests that we had to develop during this period to control the quality of our RNA products. So related to the integrity, purity, potency, safety of the RNA, which is our drug substance, and also the release tests for the drug product, the formulated encapsulated RNA. And talking a little bit more about our work, Professor Guilhermina already told you about the program from WHO that selected some hubs in different countries over the world. And our group in Oswaldo Cruz Foundation was selected by WHO and PAHO to be one of the centers for the development and production of mRNA vaccines in Latin America. And the selection was a result of a global call to increase production capacity and expand access to vaccines in the America. So we are the center in Brazil, but there is also a center in Argentina, which is Synergium. And we are working all together to get this platform being implemented in low and middle income countries. And part of the work that I already showed you during my presentation was done because of this selection. So in the beginning, we started working with self-amplifying RNA. We still work with self-amplifying constructs in our group because we believe that this will be the next generation of RNA products. We started work with constructs bearing 2 different antigens of SARS-CoV-2, trying to skip the problem of the appearance of new variants. But at certain time, we realized that it would be very difficult to register a vaccine using self-amplifying RNA because it's not still well accepted by regulators. There is only one vaccine based on this technology licensed in the whole world. It's in Japan from the company Arcturus. So and also because self-amplifying RNA is much more difficult to deliver. So we start, and also as a way to dominate the platform, we wanted to really know about this platform to profound understand what were the hurdles of this technology to move forward to other projects using RNA. So we are still working on the improvements of this self-amplifying RNA, and now we have very, very good results with this platform. But at that time, we started working also with mRNA. And some of the things that we've done during this period, I already told you during my talk. So our strategy was to skip as much as we could all the patents, or at least the most important points that were addressed by the patents of big companies like Moderna or Pfizer. So we started designing our own 5-line, 3-line UTRs. We've performed the bioinformatics work with spike sequence to modify that, to improve this translation, and also we designed a particular poly-A tail. And also we started using pseudoridine, as I told you, and we found freedom to operate lipid to use in our lipid nanoparticle. So during this, I'm sorry, I forgot to put it in English, but during this period, we've tried more than 20 combinations of ionizable lipids and constructs, totally developed at Biomanguinhos by our team. And part of this work I showed you, comparing 3 different ionizable lipids. And we found a candidate for COVID that COVID-19 is our proof of concept. It's the most advanced vaccine we have in our pipeline. And this, as I showed you already, we found a candidate that reduced the viral load, gave us 100% protection, robust T-cell response, and high neutralizing antibodies titers. So we moved forward with this candidate, and because of this work of skipping of all these patents that we could skip, we've developed and filled our own patent that was filled in December last year. So December now, starting today, we will move forward to the PCT and to fill this patent in other countries. And because of this, me and my colleague, Dr. Ana Paula, we won this intellectual property and public interest Dennis Barbosa Award. And during this time, besides developing the candidate, we also equipped and developed our own, the first RNA GMP facility in Latin America. The manager of this facility is here, Carminha. And we are very proud to have this facility in Fiocruz. And for the RNA drug substance production, we are adopting this quantum system. Quantum is a Belgian company what was financed by Gates to develop a system that would be accessible to low and middle income countries. So this equipment has a lot of technology with these robotic arms and whatever, but it's very easy to operate, and you can, and there is no need of water or gas or other utilities. So it's easy to put this machine inside the C-classified room and get the RNA produced in one day. And the encapsulation process was wholly developed by our team in Fiocruz, and now is being improved by Carminha's team in pilot plant. This is the GMP encapsulator, and this is the first lot we have produced in this facility in November last year. This is the naked RNA, the drug substance produced in this quantum machine. The encapsulation process, it's already in 4-liter scale. For RNA products, it's a very big scale. It's more or less 50,000 doses of vaccines. The fill and finish process and housing, this lot was used to perform the toxic studies, and now we are producing a lot of lots to validate the sterilizing filtration. This is the result, the partial results of toxic studies, but we already finished this test, and there was no signs of toxicity and mortality in the animals, so we are preparing our documentation to move forward to phase 1 in the beginning of next year. So during this time in Fiocruz, we were able to set up the whole platform for RNA products development and production. Since the early development with the antigen design, the RNA backbone we've patented, and the formulation development, we passed the proof of principle, producing pilot scale, performing non-clinical lots and animal tests. And we are now in this phase, performing the scaling up studies, process improvements. We are finishing the QC methods development and documentation and preparing to have the GMP production of clinical batches in the beginning of next year. And of course, when we talk about RNA, we are not talking only about preventive vaccines. It's just the top of the iceberg. We are just starting. So there are a lot of news in newspapers and sites talking about the possibilities that we have with mRNA. For example, this one is therapeutic RNA vaccine to treat pancreatic cancer, and also this personalized CRISPR gene editing therapy that uses RNA as a vehicle of this CRISPR gene. And this is a slide from a market report showing that there is a trend to increase the number of prophylactic vaccines and therapeutic vaccines using mRNA as a vehicle in the next 10 years with the increasing trend for therapeutic area also. And in Fiocruz, we started working also following this trend to be updated with the technology. We started using our patented platform to try to develop also therapeutic molecules. So we are using PD-1, PD-L1 axis as a proof of concept of using our platform to obtain monoclonal antibodies in vivo. So the purpose of this this use is to inject the RNA, the encapsulated RNA, by endovenous in endovenous way, and this lipid nanoparticle has a tropism for the liver. As you can see here, this is our nanoparticle, our result. It will go to the liver, and the liver will be the factory of these monoclonal antibodies. So the RNA will enter the hepatic cell. The hepatic cell will start producing the monoclonal antibody, and it will be delivered to the blood, and it will will perform its role in the place that it will be necessary. So this is the proof of concept of using our platform to express monoclonal antibodies. We performed an ELISA, coating the plate with the PD-1, and putting to react the hex supernatant transfected with our product, the encapsulated RNA encoding the monoclonal antibody. And we've tested two different proportions between high chain and light chain. So these are the results. So we were able to detect the monoclonal antibodies from translated by our RNA, and this is with lipofectamine, and this is with our LNP. So it was possible to see this RNA being translated in the desired monoclonal antibody. And we've performed also a test to see if the antibody would be able to attach to the PD-1 in the surface of T cells, and it was possible to see that this antibody encoded by our RNA was able to attach to the PD-1 in T cell surface. And we are also testing our platform to develop therapeutic vaccines. So as you may know, the main targets for the development of therapeutic vaccines is to use neoantigens, but neoantigens are very expensive to identify, so it will not be accessible to our unified health system. So we started work with tumor-associated antigens. So those are antigens that we could identify before and have a range of these antigens that we could use in a therapeutic vaccine that could be more accessible to our unified health system. So we are using these universal targets, put in our platform, and try to cross this valley of death. So we've performed bioinformatic pipelines to identify some of these antigens, and we were able to identify 8 tumor-associated antigens using breast cancer as a model. So you can see here that there is a huge difference in the expression of these tumor-associated antigens when we compare tumor cells and normal tumor biopsies and the surrounding normal tissue. And of course, we are going to put these antigens in our platform and test to see how it works as a therapeutic vaccines for eliciting immune responses and making the cancer to regress. So as a form of, as a way to be sustainable with the platform we are implementing in Fiocruz, We are developing a whole pipeline of products based on mRNA products. The most advanced one is COVID-19, because COVID-19 is a proof of concept. We know how it works. We know what to measure. But we are also developing vaccines against Oropouche fever, which is in an emergency in our region now. Leishmaniasis, RSV, monkeypox, tuberculosis, yellow fever, Zika. We are working in stability improvements in monoclonal antibodies expression in therapeutic vaccines, and more recently we joined an initiative that was approved by high impact financing by MRP related to the development of CAR-T and CAR-NK using RNA. And of course, as Fiocruz, during this time we have performed a lot of partnerships. We've trained professionals from Cuba. We have a lot of relationship with other units of Fiocruz in Brazil. We have relationship with Europe companies and universities, with UNSW in Australia, with other members of our initiative, WHO, with Synergium, and we are being financed by Bill and Melinda Gates through this RNA network initiative to prepare our knowledge to be transferred to other countries, mainly in Africa and Asia. So our goal, of course, as a Fiocruz unit, is to increase the capacities in this area in our region and also in Africa, and we advocate for equitable access to these cutting-edge products across our region. And so I would like to acknowledge all my team in Fiocruz, but also all the people in Biomanguinhos who are involved in this work, people from pilot plant, from other labs, from quality control, a lot of work being done. Of course, I'm not able to develop all the things that I have shown here, so we have a lot of people very committed to make this work. So thank you so much.
So thanks, Patricia, for the nice presentation. Do we have questions?
So thanks for your presentation. So you have said that the PA ball is the best nanoparticle to use as a delivery system, but on the other hand, it's also the worst nanoparticle to induce immune response. Could you explain me again why this happens? I think that I didn't get it.
Because for preventive vaccines, there is an important role of lipid nanoparticles. So besides delivering the RNA, the components of the lipid nanoparticle also elicits immune system. So for example, if we can compare, we could use PA ball to deliver a therapeutic RNA. For example, to replace a protein that's missing in like a genetic disease, because it will deliver very well. You have a lot of expression of the protein, but it will not elicit immune responses. But for preventive vaccine, it's the opposite. Sometimes you don't have the highest expression of delivery and expression of the RNA, but you have the role of lipid nanoparticle eliciting the immune responses. Do you understand?
Understand.
Okay.
Thanks a lot for your presentation. Awesome work. We're all very proud of Fiocruz, being Brazilians, right? And I got really curious about the mAb application, producing mAb in the patient using messenger RNA. And I mean, let's say you have a commercial mAb product, but then you just produce that mAb inside the patient somehow using messenger RNA, for example. Is that something that would be like a break of patents? Like if a company has a patent to produce a mAb, that's a drug product, but then you produce the same molecule inside the patient's body, what's the conflict like? Is it a new therapy? It's not a product? How does it work?
From my point of view, it's a new product, but I don't. It depends on what. I think that Ana Cláudia could answer better than me.
It's a new product.
Yeah, I think
Yeah,
that
it's a new
if
product.
the sequence is not protected,
Yeah.
you just can use, and it's a new product.
Yeah, if you all the steps not protected, it's a new product from them. Yeah.
We have still work to do on it because we need to adjust the doses because you are not injecting the final product. You are injecting something that will turn in final product. So we have some steps to advance to understand how it works, the mean concentration it will achieve in blood during how many time, things like that. But I think that in the future it will be an alternative, a cheaper alternative to use monoclonal antibodies.
Any more questions?
Thanks for the excellent presentation, Patricia. I have a few questions here. One is, maybe I missed what you said, but you mentioned the non-amplifiable circular messenger RNA. What are the advantages of using that kind of RNA? The second point, regarding Pfizer's and Moderna's vaccines, do they use pseudouridine or N-1 methyl pseudouridine? I was curious about that. And I have still two questions. Do you want to answer the two first?
Yes. So I forgot the first one.
What would be the advantage of using the circular
RNA?
The
yeah.
circular RNA?
So this is more stable and more easy. It's easier to encapsulate, and it's not necessary to use capping, and it lowered the costs of production because the cap analog is one of the most expensive components of this production, and it also improves the translation of the RNA. So it's still in the beginning of the development, but it's a promising new technology related to the RNA platform. And the second one, yes, Moderna and Pfizer are using N1 methyl pseudouridine. It's patented by Moderna. They are fighting because of that.
So I would be interested in hearing from you about duration of immune response, because I think nobody nowadays doubts that mRNA vaccines are a very, very, very good tool for pandemic responses. But regarding long-term or child vaccination for life and so on, do you see RNA vaccines having a durable immune response, or maybe mRNA vaccines will be pandemic vaccines, and maybe recombinant subunit vaccines will be childhood vaccination tools, or so on?
I think that a lot of researchers are trying to address this issue of mRNA platform. I think that self-amplifying RNA will be a solution because it's much more immunogenic than the conventional one. And also there are some people trying to put adjuvants in these components of the lipid nanoparticles. So there are a lot of people trying to improve this memory formation. By now, I think that this is a very good platform for pandemics and also for therapies. We are discussing a lot the use of RNA for therapies, but for this vaccination for children, and maybe we need to improve the memory formation.
And now the fourth and last one. When we think about large-scale production of mRNA vaccines, you showed that we need the plasmid DNA as the template for RNA in vitro transcription. Is the scale of plasmid DNA that we need big? And if it's big, is it you will need big plants of plasmid DNA to produce large scale of mRNA or not?
No, now our process is in 10-liter scale. Ludinha está aqui, pode falar melhor. And it produces a lot. Do you want to talk, Ludinha?
Vou falar em português, tá? Então, a nossa escala de produção do DNA plasmidial é em 10 litros. Né? Nós estamos otimizando ainda algumas condições. Né? Eu participo, responsável pela etapa de upstream, Ana Paula, que está aqui do lado da etapa de downstream, da produção de DNA plasmidial. E, assim, os resultados estão bem interessantes, né? E estamos trabalhando também na parte toda do controle de qualidade do DNA plasmidial. E, na parte da tarde, a Gabriela, que é a gestora da nossa área, vai fazer uma apresentação bem mais detalhada de todas as etapas do nosso processo. Né?
Só uma dúvida. Em qual a classificação de sala que vocês fazem a produção do DNA?
Na nossa área, a gente trabalha em BPL. Né? Nós iremos transferir o processo para a área da Maria do Carmo, que é GMP. Aí tem escala de bioreatores de 150 litros, 60 litros, em área classificada para a produção em GMP de DNA plasmidial. Né? Tanto up quanto down.
But when you mention 10-liter scale of DNA, I don't know, translates, let's say, X grams or X milligrams of DNA, and this will then translate into how many doses of a typical mRNA vaccine?
Well, nós estamos otimizando a etapa de purificação e estamos obtendo uma massa bem maior de DNA plasmidial, né? Estamos obtendo em torno de 200 miligramas, mas com o processo que a gente está usando de TFF, né, e com o equipamento, o ACTA, etc., nós estamos conseguindo, melhoramos esse quantitativo. Né?
E isso daria quantas doses de vacina de RNA? How many doses?
For each 5,000, for one lot of that I showed here, 5,000 vaccine doses, it's necessary 15 milligrams. 15 of plasmid linearized, 15, 15 milligrams of linearized RNA. So it's almost 10 lots we can have with this yield. Yeah.
So I have a question regarding the logistics to distribute mRNA vaccines. So how are you planning to develop your vaccine? Should it be frozen or liquid or lyophilized? How are you planning?
It's liquid and it will be frozen. By now we are using the traditional process. Stabilizer is sucrose, but now we are working with an England company that developed a thermostabilizer. We are testing it, and with the Pfizer's formulation, they were able to show that it would be stable for 1 year to 2 to 8 degrees. So we are working with them in this technology, never frozen, that Pfizer is also using now. So we are also working these thermostability issues.
So any more questions?
Hi, thank you for your presentation and congratulations for your work.
Thank you.
My question is about the in vivo map production, just like Ruiz. I was curious if aggregates and conformational variants of the antibody is a concern in your technology of producing it in the patient, just like it's a key point of interest in the production of mAb as mAb the product. And if so, how are you planning to overcome this non-optimal mAb production in vivo?
So it's less. I think that one of the things that will be an advantage to use the in vivo production is that you are not producing in a system where you are going to have all this protein like in a supernatant, and it will be favorable to aggregate. So when you inject the RNA and the mAb is produced in vivo, you are producing your own protein. It's like an antibody you produce in your immune system. So with your glycosylation, with your amino acids, with your machinery. So one of the advantages to use the RNA in this case is to minimize the reactions, the allergic reactions, and I don't think that we hope to have this problem of aggregation using the RNA.
Regarding costs, comparing mRNA versus antibody, how do you think it's going to be comparable?
So I don't know the cost of the therapies because the doses will be higher. But for example, for vaccines, our target is at max $10 a dose. So if we multiply this per, I don't know, 10 or 20, it will be still cheaper than the monoclonal antibodies produced in CHO.
Okay. Another question. Regarding, if we think about health sovereignty, the future pandemics that we will have, regarding raw materials for producing the mRNA vaccines, are we depending on imports? Are there maybe any tools that Brazil and other countries could use to motivate companies to produce locally the raw materials? Because we must have the technology, but we must have the raw materials as well.
Yes, yes. This is a big problem for all of us. But we are working in this project to bring at least some of the raw materials to be produced in Latin America. So we are discussing with Argentina and Parro to have the lipids produced in Argentina. And we also have an initiative with IBMP in Paraná to produce the nucleotides and enzymes. So we hope that we have in a short period, I hope, part of this material produced here. But we are still depending on plastics, all these things that it's difficult to bring, companies to produce here.
Okay, so thanks, Patricia, for your presentation. It was a really nice one. So with this, we close our morning program for today. And
we have some information to give. Sorry. So just reminding you, as I said before, you must sign up for the workshop you want to go to. Please do it now or until the end of lunchtime, so until 2 p.m. After 2 p.m. we will close the lists, okay? And then when you go out here after the next lecture at 3:15, we will be showing outside the rooms for each workshop, okay? Because the room choice depends on the number of people in each workshop. We also wanted to remind you that no food and no drinks are allowed in the conference room, okay? We want to keep it as beautiful as it is now. And also repeating again, no pictures of presentations, please, okay? So we will start punctually at 2 p.m. again here. So we delayed a bit with the coffee break, but we will recover and be again in time in the afternoon. So be here sitting down at 2 p.m., please. Thank you. Speakers, please gather here for us to have lunch.
Chegou, fez não sei o quê, dobrou